Hi, everyone. I'm very happy to be here as part of the SCN2A virtual conference. My name is Jennifer Tjernagel, and I am the project manager for Simons Searchlight as part of the Simons Foundation research cohorts. Today, I'm going to tell you a little bit about our SCN2A Research Registry that we have in Simons Searchlight. Okay, so overall, what is Simons Searchlight? It's a research program, and our goal is really to accelerate the genetic science behind these neurogenetic conditions associated with autism and other neurodevelopmental conditions. So SCN2A is actually one of our largest groups. And at the end of the day, we want to shed light on all of these conditions by both collecting high quality natural history data, as well as really building strong connections between researchers, families, and industry partners. So there are a few elements to this I want to highlight on the screen. First of all, we collect the detailed medical and behavioral histories, as well as blood or saliva samples for our biorepository. And we make these available to all researchers. Secondly, we feel strongly that we not only want to collect data, but we want to provide information back to participants. So we synthesize this information that you provide, and we share the results back to you. There are a few ways that we currently do this, but we're always looking for new ways to provide valuable information back to the community. As far as sharing the information more widely, that's one of the core principles guiding all that we do here at the Simons Foundation. We believe in broad access to this information. So we freely share the data and biospecimens with qualified researchers. And we actually have an entire portal devoted to sharing resources with the external community. And this is to any researcher around the world. I'll talk a little bit about that later. Okay, so as far as what we collect in Simons Searchlight, I know this is a busy table, but it really just outlines what we collect and how, who we collect information about, and how many times. So the first item I want to point out is at the top here under medical records. One of the first things we ask for is the clinical genetics lab report that spells out all of the genetic diagnosis information. This is really to make sure that everybody ends up in the right place and that we can extract information from that report that details the genetic finding and the information really necessary for researchers to do their analysis. So we collect that on both individuals with the genetic condition as well as any other family members that have had genetic testing done. Next, we conduct a medical history phone call to get some additional information. So in addition to the medical history, we collect information on diagnoses and ask about medication use. The medical history interview, as well as the diagnoses and medication, is all done at the initial time point as well as over time. So when we say longitudinal, that's roughly every year on or around that person's birthday. We'll check in and get updates to put back into the registry. The online surveys that we have here at the bottom of the table are varied. We're just adding a sleep survey right now. And we have detailed seizure history survey, adaptive functioning measure called the Vineland, as well as other standardized questionnaires and some surveys that were actually ideas from participants that led to the development of these surveys. And the skin and dental and the learning to read surveys are examples of those. Currently, we are available in English and Spanish, and we're going to be adding new languages by the end of this year. French, Italian, and Dutch are next on the list. As far as our biorepository, we collect blood and saliva in some cases. Our blood collection system in the United States is with Quest Laboratories, which is a network of labs all across the country. We send a box of tubes to the participants' homes that they can take to their local Quest center to get those that blood drawn and then ship it back to us. In other countries like Australia or some parts of the EU, it's a little bit harder to get the shipping coordinated in the right fashion. So we're currently looking into other options there. Okay, so giving back information to participants is really high on our priority list. We have standardized reports that we will provide back for some of our surveys, and that's the example that's shown here. On the right, this is for the Vineland, which provides information on daily functioning. The report explains a little bit about what this means and shows results for, in this case, the SCN community as a whole of who we've collected and where your particular child falls in those ranges. In addition to the individual results reports shown here, we also do summary snapshot documents that look at the entire community, SCN2A, for example, and provide high-level information in a visually easy to understand way that we can then send repeatedly over time. So we're currently working on doing this quarterly, and we'll be launching this at the end of this month with our first version, so stay tuned for that. The presentations that we do are very similar to the snapshots, and sometimes you will have seen some of our PIs or co-PIs give these presentations. So we either have these in virtual mode, like we are now, or in the days when we actually were able to get together in person, and we hope to be able to do so again soon, we'll actually travel to conferences and give presentations to outline the information we've collected through the registry to date. Along with sharing back with participants, we want the registry data out in formats to contribute to ongoing scientific knowledge. So this includes genetic reference guides, such as ClinVar and gene reviews, and scientific journals and conference presentations. As far as sharing data and biospecimens with the research community, as I said, we have an entire portal designed to do this for all of the cohorts that we manage at Safari. The portal is called Safari Base. And this is the way that external researchers can come to the site and apply to use any of our resources, including data that we collect through surveys and phone interviews, genetic data, and the samples in the biorepository. We also have a way for external researchers to recruit Searchlight registry members and send additional surveys called Research Match. And I'll talk a little bit more about that in a few slides. As part of the growing biorepository, I want to highlight a new effort that we started in the past year. We're generating a resource of centrally created induced pluripotent stem cells, or IPSCs. This is a special cell type that researchers have been very interested in accessing. There's a lot going on in this particular schematic, so I'll walk through it. Once we get a blood sample from an individual, we can use some of the components in this blood to generate these IPS cells, which basically are very early versions of the cells before they differentiate. Researchers can then use methods to coax these cells into more specialized cell types, such as muscle cells, cardiac cells, or in this case, the interest is really into brain cells or neurons. From using these neurons, they can also be called mini brains if they've been grown for a little bit. They can generate hypotheses, do disease modeling, drug screening and discovery, and having these cells available to order from Simons Searchlight will save on time and cost, and it's an important part of a toolkit for researchers. So we're really excited to get this going. All right, I said I was going to talk a little bit more about our Research Match program. This is instead of sharing information, only the information that we collect as far as data and biospecimens to the research community. This offers a way that they, the external researchers, can collect additional information that they're interested in. So if they have a more detailed survey that they would like to collect, for example, on the SCN2A community, or they want to recruit externally for another study that they are running, including potentially a clinical trial, for example, they can use our Research Match program to have this built into our system to automatically send to all of our participants. So here are some examples of activities in SCN2A that collectively show the power of Searchlight and the Simons Foundation efforts over the past few years. We funded 9 research projects over 8 research groups specific to SCN2A. We'll see that on the right. We funded the creation of an SCN2A rat model that's now in our repository at Jackson Labs and available to researchers, as well as 6 IPSC lines that are in progress that will be available to order soon. We were able to provide data and information to collaborators such as Ann Berg at Lurie Children's Hospital in the US in support of a recent SCN2A publication using the Simons Searchlight data. And we worked closely with Catherine Howell at Murdoch Children's Research Institute in Australia to help recruit for their natural history study. And we'll be working with her group in the near future to get that data added to our repository as well. So those collaborations have been really successful and we've enjoyed working with them. We have had 39 individuals with SCN2A who have provided biosamples and 6 different research groups that have been accessing and using the Searchlight data specific to SCN2A. As far as the research match component that I just discussed, you may have seen several COVID-19 surveys come through in the past year, and we've published those results. And we have several other projects in the works that help recruit and collect data for other external researchers. And most importantly, across all of these efforts, the true power of Simons Searchlight comes from individuals and families who continue to dedicate their time and knowledge to participate in this effort and provide us with feedback. We have a community advisory committee of 20 members at this point that shares valuable insights with us on what's most important to the Searchlight community, and we're really thankful for them. So where are we with SCN2A participation? We have over 200 families that are registered. From there, after initial registration, then individuals will fill out their consent form. They'll upload their genetics lab report, complete their medical history call, as well as complete other surveys. So you can see over time in this graph that there are people in different stages of participation. Our current focus, at least for the months of February and March, we're focused on really trying to get individuals to locate and upload their lab reports. So you'll see in this graph the difference between 173 individuals who have consented and 133 who have actually uploaded their lab report. So we're really trying to get those additional pieces of information so we can keep moving those participants on in the process. If you don't have a copy of your lab report, our study team can request it on your behalf and provide it back to you after we receive it. We just launched a digital medical release form to help with that so that you don't have to be providing a paper authorization to get those in your behalf. So to see where you are in the process, whether you think you still have something to do or you just want to double check that you've done all the steps, you can go to your study dashboard or directly contact our study coordinators. I've got the email here, so please reach out if you need help. As I said, we are an international registry and we understand the incredible need in rare disorders in particular to really band together to increase the number of people under study to really accelerate the knowledge into these conditions. You can see from this table, we have individuals from around the world. And although we've started in the US, we have a growing number of families in other countries, and we aim to significantly boost that in years to come, especially with adding other languages. The numbers here represent only the SCN2A community. So as an example, we have close to 50 participants from Australia across the board in Simons Searchlight, and 7 of those are from the SCN2A community. Okay, this is a list of the genetic variants that we currently see in our SCN2A Searchlight community. This includes pathogenic, likely pathogenic, and variants of uncertain significance. The main message here, I know there's a lot of information, but there are a large number of unique SCN2A variants in this community, and the importance of individuals needing to represent themselves in research. You just want to make sure that you're represented on this list when researchers come to us for data or samples. I'm going to show a couple slides here at a very high level just to describe the participant community as data we've collected to date. Right now, we have individuals from age 6 months up to 56 years that are currently participating. And when you read this graph, you'll see the height of the bar indicates the number of individuals that fall in that group. So for example, we have 31 individuals that fall between 0 and 4 years of age. Overall, you can see this is a young cohort, but it's nice to see that participants are also in the upper ages as well, because it's very important for the whole community to assess the trajectory of what SCN2A can look like over time. And there's a lot to learn from all the age spectrum there. As far as developmental and behavioral diagnoses reported, this is based on 77 participants that responded. You can see that 67 of them reported developmental delay or intellectual disability. So it's a very, very common in the SCN2A registry. The next would be autism and language impairments, and to a lesser extent, other diagnoses are represented here as well. For neurological conditions, this graph is based on 83 respondents, and you can see a very high percentage of individuals reporting low muscle tone or hypotonia. Next, most common would be individuals that their parents report as being a bit more clumsy in general, and also individuals that have been officially diagnosed with a movement disorder. We also see high muscle tone in addition to the low muscle tone, but then several other conditions such as a larger head size or a smaller head size than average. One individual had a cyst in their spinal cord. We have several individuals with tic disorder, cerebral palsy, or cortical blindness. For seizures, based on 83 participants, and in this graph, as well as the previous graphs, I'll just point out that individuals can be represented in more than one category. So they could have had more than one seizure type and be counted. You can see a higher level of grand mal seizures and infantile spasms reported, but other seizure types are seen frequently as well. We do ask respondents if there happens to be a particular medicine that seems to work best for their child. And while not all respondents identified a best medication, two identified to be working well are valproic acid and Keppra, or levetiracetam. So in summary, some of the common issues or issues we want to point out were developmental delay, cortical blindness, or other vision problems. Seizures obviously continue to be a significant problem. Low muscle tone, and then not shown on the slides from today, but we also see GI issues and ear infections that rise to the top there. Less common but notable issues include autism spectrum disorder, language impairments, intellectual disability, movement disorders, and then again, not shown on the slides from today, but we also see scoliosis and endocrine issues. Okay, so for any researchers online, if you're interested in accessing this information, that I've shown you the high level summary of the data, but if you go in and actually order the data files, there's much more granular detail in there to dig into. We have the common portal that I mentioned called Safari Base, and the web address is here, but it has all the information you need to go in and request the data and biospecimens. And again, we have a whole team that's dedicated to distributing these resources. So to join Simons Searchlight, if you haven't yet joined, or if you've joined and haven't yet had the opportunity to complete all the steps, it's fairly straightforward. I'll just walk through these here. You sign up online, you give us your genetics lab report that I talked about. You have a medical history phone call with our genetic counselor, then you can fill out surveys online. And if you're interested in providing a biospecimen, you can do that. And importantly, follow up with us over time so we can check in and see how everything's going and collect information on a yearly basis from you. So overall, why is it important to join research in general? In addition to needing more numbers for researchers into these research into these rare conditions, as I said, it's important to represent yourself in research because the effects of these genetic variants can be different. You just want to make sure that researchers have their eyes on your particular experience and really round out the picture of what is being experienced in SCN2A as a whole. And as far as the sense of urgency, why it's important to do right now, the message continues to be how important it is to get your community ready. So having a research toolkit that includes natural history data, cell lines, mouse models, appropriate outcome measures, et cetera. If you have these components ready, you'll be in a much better place to move forward as quickly as possible when opportunities for therapeutics arise. Okay, thank you again for including me in this conference, and I look forward to hearing from all the other speakers.